Welcome to this biochemistry video on DNA technologies. So DNA is the biopolymer that nature uses to keep track of genetic information, and we've hijacked that a little to improve our lives, basically, and for applications in biotechnology. And that's what we're going to discuss today. So PCR is probably the first and foremost DNA technology that we use. It's used to amplify DNA in a test tube. So the basic steps are pretty straightforward. We design primers that will bind to a region of DNA. We dehybridize the DNA by increasing the temperature to 94. We then re-anneal the temperature, or re-anneal the primer and the template strand of DNA. And you can see that in the second step, and that will be the start of, and that temperature is around 68. That depends on a few factors. And then we raise up the temperature again and allow for elongation, where the polymer, or where the polymerase comes in and finishes making copies. And we do that over and over and over and over, and at the end of the steps we get very large numbers of DNA, of the same simple DNA. Again, the three steps are denaturation, where we take and we heat up the sample, and the two strands dehybridize. Annealing, where we anneal the primers to the two template strands, and then elongation, where the polymerases come in and make copies. And then again, we do this 25 or 30 times, and voila, we end up with large numbers of the DNA. You can see if we do it 30 times, we end up with billions of copies of the same strand of DNA. Important considerations are primer design. When we do generally put in two sets of primers, generally they're 20 to 30 nucleotides long. They're synthetically produced. We can manufacture them. We don't have to find them. They're produced complementary to the 3' ends of target DNA, and they can't be complementary to each other. And we try to avoid things like inverted repeats. They usually have to have about 40% GC concentration, because otherwise the annealing temperature becomes a little bit annoying. As we mentioned in previous lectures, the temperature, when we raise the temperature, the DNA dehybridizes, and so GC content is very, very important in determining that, because the temperature, since GC has 3 hydrogen bonds when we denature proteins, that is the lowest common, or that is the important factor of determining the temperature. And you can look up melting temperatures and calculations online. There's plenty of websites that'll tell you paste in a sequence, and it'll give you the melting temperature of a strand of DNA. Again, after we get done, we have billions of copies. We want to make sure that it worked, so we usually take it out and run it on a gel, just like you did with protein gels. There's DNA gels. We load this up, and then we spread it out spread the different size of length of DNA on a gel. The matrix is a little bit different. It's an agarose instead of a polyacrylamide, but the concept is very similar. We're going to apply electrical current through a matrix, then it's going to separate it by size. We're going to then stain it. Usually we use ethidium bromide as a stain. That's UV fluorescent, and then you put it under UV light, and you can see the bands very very clearly. That's what you see in these. So for instance, different size DNA based on the PCR results, and that's how things are visualized. A modification of PCR is qPCR, and it's real-time, or there's a fluorescent probe. There's a couple different types. There's RT and qPCR. Both are used to do quantification of the PCR process. That allows us to tell how many strands we started with. And basically we have a fluorophore and a clenching molecule, and as the reaction occurs, that fluorescent probe is measured, and that gives you a signal on how many molecules have been how many signals have been created or amplified from the original stock. So this quantification allows, this qPCR and real-time PCR allow for quantification of the number of molecules you have and gives you an idea of transcript level and other things. We also have what we call gene chips or microarrays, where we can take two different conditions. In this example, we have a fertilized egg and a tadpole. We can purify out the mRNA. We make what's called a cDNA copy of that RNA using a reverse transcriptase. The DNA then is flowed over a printed slide that has different strands of DNA on it and that are complementary genetically to different other strands. If the two hybridize, we're going to get a nice signal. So, and then we compare those signals. And so what we get is, if we have the left-hand sample is green, the right-hand sample is red. If we get a red and a green together, we get a yellow. If we have an individual green, then that means only the left condition was amplified. If we get only red, then the right condition was amplified. And these are very, very important for determining whether samples are upregulated or downregulated. And DNA microarrays were supposed to be the big deal about 10 or 15 years ago, and we have several applications of them, but they haven't taken off quite a bit as we wanted to. You are likely to see these more as time goes on, as the technology kind of matures and we get different applications. One of the very, very important things is DNA sequencing, and this was there's various methods of doing this, but the steps are pretty well the same. I mean, there's different, and this technology changes all the time. So I'm going to give you some of the historical ways that we've done this, but honestly, if you look online, you can find numerous ways of doing this. This is the Sanger method of sequencing, called pyrosequencing, and we add blocking nucleotides in there at a certain ratio, and we will get different, as we chromatographically, we will get different lengths of DNA with different signals. So C has a red signal, adenine has a blue signal, thymine has a yellow signal, and guanine has a green signal, and we add blocking in there and in a certain concentration. Blocking nucleotides, meaning that the transcript will stop at those sites at a certain frequency, and so you'll get full-length transcription, and then you'll find that the sequence will stop, and we spread these out on size, and so you can read the colors and tell what that next sequence is. That's the basic steps. This is very, very important, and things like Illumina have built on this technology and allow us to do whole genomes now within days and at a very inexpensive cost. And this is very, very critical in the biotechnology revolution that we're going to be facing in the next dozen of years. Another important technology that you're going to see is CRISPR-Cas9. This allows us to do gene editing and programming in live organisms. And so let's say you have sickle cell anemia. In the future, we will likely be able to go in with CRISPR and go in and modify those both in the germline and as an adult. And so this technology will radically change because a large number of diseases are genetic diseases. Things like Huntington's and Parkinson's and sickle cell anemia, all are going to, even some cancers are going to be able to be modified using this type of CRISPR-Cas9 DNA editing technology. And we'll show you a video. I'll post up a video that I really like on this technology. I don't do it justice. The videos that exist on this are very, very excellent and elegant. One thing that we'll probably see a little bit more greater detail in a different video is restriction enzyme cloning. This is where we take, and there are enzymes that exist that clip out DNA at certain sequences, certain sequence patterns. So GCC-GGC is a site where endonuclease called BSR-F1 will clip, and BSPE1 is the site is TCC-GGA. So there are enzymes that will clip DNA at a specific site in a certain pattern, and we can use that to clone. And cloning basically means we take a gene from one organism and put it into a controlled system of another, and generally we put this into plasmids, which are circular strands of DNA, and we can put in certain genes, and we can produce, for instance, in this example, we're taking spider silk and putting it into E. coli, and that allows the E. coli to be put in, and then we ligate in that gene, and based on the compatibility site. So we make a compatible strand of DNA and put it in there based on those restriction sites. Again, very fascinating technology. We'll post something that will give a little greater detail on that. It's very, very important for future work, and cloning allows us to make synthetic constructs and provide also things like genetic resistance for applications in agriculture. Thank you for listening. We look forward to the next one.